Welcome. Good afternoon and welcome to the February program of the National AgTech Forum, hosted by the North Carolina Biotechnology Center in partnership with AgStart and AgLaunch. I'm Michelle Von Cannon, the Agriculture Program and Event Manager here at North Carolina Biotechnology Center, headquartered in Research Triangle Park, North Carolina. The Biotech Center and its Ag Sector Development Team are committed to leading life science economic development for our state while promoting the ag tech industry by supporting innovation, commercialization, education, and business development. You can learn more about our programs and funding opportunities by visiting ncbiotech.org/agriculture. We are excited for today's program. It's the second one this year in 2021, titled The Cat in the Ag, CRISPR, ARCUS, and Talons. But before we get started, I'd like to offer a few housekeeping notes. Today's program is being recorded and will be posted online in the next couple of days, along with the PowerPoint slides from the presenters. You'll find that on the event page on our calendar at ncbiotech.org/events. Make sure to insert your questions in the Q&A tab below, as there will be Q&A at the end of the program. Also review the other questions that are listed there, and if you like one and want to promote it in priority, give it the thumbs up and like it. Please reserve the chat tab at the bottom for questions that we will give to the audience and any technical issues you might have and would like us to help you with. Also following the program today, we are going to have virtual networking. Our speakers have graciously agreed to stay and host a virtual room based on their specific technology. Additionally, we'll have 2 general networking rooms hosted by 2 of our regional annual sponsors, Smith Anderson and Hughes Pittman Gupton. You will be able to freely roam in and out of each of the rooms as you like. On that note, technical difficulties. I'd like to thank our sponsors, Global Ag, Smith Anderson, Myers-Beagle, and Hughes Pittman Gupton. Those are our regional annual sponsors, as well as our regional supporting sponsor, Alexandria Launch Labs AgTech. So with that, I would like to introduce Rod Wilson of HPG. He has a few announcements to share with everybody.
Thanks, Michelle. Good afternoon, everyone. As Michelle mentioned, I'm a partner with Hughes Pittman and Gupton. We're an accounting firm located in RTP, North Carolina. We're a full-service accounting firm that provides tax, audit, and outsourced accounting solutions. And with a focus, we're in a variety of industries with a focus on life sciences industry. And I lead our ag tech practice, which is a subsegment of that life science industry. Before we get into the program, I'd like to draw your attention to a couple of upcoming events. On March the 15th is the monoclonal antibody therapy and veterinary medicine event, with more details to follow, available on the ncbiotech.org website. And then on March 17th is the next iteration of this National Ag Tech Forum, entitled Farming, What's Next? Unlocking the Potential of the Soil. So please join Farm Journal's Margie Eckelkamp and National Black Grower Council's Dr. Dwayne Goldman for a discussion around soil health data and carbon capture with entrepreneurs Rebecca Carlson of Nori Carbon Removal Marketplace and Mitchell Hora, a 7th-generation farmer and founder of Continuum Ag, quantifying soil health data. So we look forward to seeing everyone at those upcoming events. And now I'd like to turn it over to my colleague and co-sponsor of the series, Mike Saber from Smith Anderson. Mike.
Okay, thanks, Rod. Welcome, everybody. I'm Mike Saber with Smith Anderson. We're a law firm headquartered here in Research Triangle Park, North Carolina, where we from where we serve our clients around the world. We have a global footprint. We're one of 9 law firms in the world to be ranked by Chambers Global, recognized as one of the preeminent firms with an agribusiness law practice. I'm excited about today's program, and I have the honor to introduce our host and speaker, Joerg Bauer. Joerg has 20 years of global experience in bioindustry. He's held leadership positions at BASF and Metanomics, which is a BASF affiliate. Joerg has a background in plant physiology and plant biochemistry with a master's degree from University of Hohenheim in Germany and a Ph.D. from ETH Zurich in Switzerland. His responsibilities at BASF and Metanomics have included academic and industrial collaborations, global project management, and successful product development. He's the author of numerous publications and the inventor on multiple patents. Joerg is currently Global Head of Scouting and Collaboration Management Bioscience Research at BASF. In this role, he connects technology innovations of basic and applied research from universities and startups with business needs of BASF to enable the development of new products. Joerg and his family have lived in beautiful North Carolina since 2011, and we are pleased to have him here to host our program today. Thank you, Joerg.
Excellent. Thank you very much, Mike, for this kind introduction. And we really love it here in North Carolina. There is no snow here. So for everybody else who has snow, great to have snow, but also great to have not snow. So it's a pleasure to be here today and moderate the forum. We have 3 exciting guests today, all 3 of them captains of gene editing, navigating the blue ocean of agricultural trades with their respective companies. So we have Rodolphe Barrangou, co-founder and CSO of TreeCo. We have Travis Frey with Calyx, Chief Technology Officer, and we have Fayaz Khazi from Elo Life Systems, CEO. So we have quite an interesting topic, the cat in the egg, gene editing applications in agriculture. So you heard about gene editing. This is one of the most exciting scientific stories probably in this decade. And just a couple of pictures here to break the ground. You know about the Nobel Prize from Carpentier and Doudna last year. There is actually, I want to turn your attention to a movie about gene editing, Human Nature. Many of the key players, scientific key players, are broadcasted in that movie, actually also Rodolphe. So if you ever want to know the number plate on Rodolphe's car, watch this movie, hopefully not only for this. There is exciting movements in agriculture with this technology. You see here the quote from Kevin Folta, and you probably also heard from USDA, the secure rule, where they are actually adjusting the regulation needs for gene-edited agricultural products for croplands. So it's all exciting topics, and that's what we want to elucidate today in the forum, and look a bit deeper, look under the hood, what's going on. We have 3 areas: technology, business, and customer regulatory. So this is what we're going to do in the panel discussion. Before we go into the panel discussion, each of our 3 guests will give a 10-minute insight into their respective companies. And after that, we will go into the panel discussion. We have prepared questions, but of course, you can also start filling up the question and answer box with your questions. We will have then latest on 5:15. We will start with the questions from the chat box. Will we see? Maybe there are already questions which can go in earlier. And then, as Michelle introduced, at 5:30, we will have the breakout rooms where you have then again the opportunity to get further questions answered or use it for networking with one of the 3 guests today. So very exciting. And I'll just see we have 145 participants. So hopefully we meet 150. If you know somebody, forward the link. It's still time. We haven't started yet. For the first presentation, I want to introduce Rodolphe Barrangou. And Rodolphe is the distinguished professor, Dr. Clenhammer Professor in Probiotics Research at North Carolina State University. Rodolphe is very well known for his characterization of the CRISPR-Cas systems and its applications in bacteria. Rodolphe has spent time at DuPont very successfully. He decided then to go to NC State, and actually in a recent panel, he gave a little bit background. So he decided to go to NC State to also have the freedom to found companies. And actually that is what he did. Rodolphe is co-founder of many companies, Intellia Therapeutics, Locus Biosciences, CRISPR Biotechnologies, Ancilio Biosciences, and TreeCo. So TreeCo will be the company he's presenting in a minute. Rodolphe is also elected as member of the National Academy of Sciences, the National Academy of Engineering, and the National Academy of Inventors. So quite a number of things. Rodolphe, we are happy to have you here, and please give us some insights into TreeCo.
Great. Thank you, Joerg, and it's great to be in North Carolina indeed. All the years I spent at DuPont, I spent living in Wisconsin, and I shoveled for a lifetime, and I'm happy that we're all here today. And those of you who have to shovel today or tomorrow, good luck. So indeed, genome editing is arguably the technology of the decade, as just mentioned. And I'm here in my capacity as a co-founder and the chief scientific officer of TreeCo, a North Carolina tree company, a startup out of NC State. And our goal is really to combine insights from tree genetics with the power of genome editing to breed a healthier and a more sustainable forest. And as a matter of fact, as you can imagine, with climate change and global warming and issues that we have regards to sustainability for the planet, we have our work cut out for us. And in order for us to address those grand challenges, unequivocally, we need to be able to deploy great technologies. And today, I look forward to talking about genome editing, whether it's CRISPR or ARCUS or Talon. Genome editing, especially with the collage that Joerg just presented to us, is unquestionably or arguably the most timely, the most compelling, the most promising technology. And if you look at the science, and if you look at the business, and if you look at the technology and the investments, 200,000 plasmids have been distributed to not-for-profits by AgTech. I mean, it's a tremendous number, and the growth is very, very compelling. If you look at the scientific literature, again, thanks to meganucleases and ZFNs and Talens and ARCUS, planting the seed for genome editing early on, the rise is compelling on a log 10 base here. And this is just the beginning, arguably, of the CRISPR and genome editing revolution. And it is global, whether you look at authorships with the US #1, China #2, but as a matter of fact, 5 of the top 10 most prolific nations in the CRISPR literature are in Europe. Even with the UK coming out of the EU, they are physically in Europe, and we have obviously the Americas and Europe and Asia and Oceania representing the top 10. So this is democratized, this is globalized, this is widespread, and this is just the beginning. But if you really look at the business landscape, we see that notwithstanding the relatively young age of genome editing, in the CRISPR startup landscape in particular, we're already looking at 3rd and even 4th generation startups coming out. At the very top, the first original 2013 founded companies, most of them are on the NASDAQ today, On the right, in the therapeutic space, more and more companies coming out on a regular basis, including Beam already on the NASDAQ as well. In the bottom right, companies in the microbiome and antimicrobial space. On the far left, the tool space. And on the bottom right here, we see the ag world already, to some extent, aggressively catching up with the CRISPR craze in terms of startup. This is what TreeCo is, but also in ag and pairwise in our own backyard here in North Carolina in our team. As far as TreeCo is concerned, we're trying to deploy those promising genome editing technologies, again, to breed a healthier, a better, and more sustainable forest. And for those of you who don't know much about forestry, you may be surprised to hear it's a $700 billion global industry. It's a market domestically on par with automotive industry. And our state of North Carolina itself is arguably the wood basket of the world with $32 billion local markets. But even worse, arguably, than some of the plants that we work with, conventional tree breeding is very slow. So breeding by editing is a new approach. And I think arguably only 20 or 30 years behind in terms of molecular biology adoption. This field is ripe for disruption. And when you think about having an impact, I'm actually kind of perplexed by the relatively small number of people who work on trees. A tree, in terms of carbon biomass, constitute the large majority of what we have here on planet Earth. If you really want to make a difference, feeding the world is great, therapeutic is fantastic, obviously, as we all know. But the ambitious goal of the company is really to revolutionize breeding of trees to breed a healthier and more sustainable forest. So this is why we're in North Carolina. I think a lot of people here online, wherever you may be pleased to hear that the Southeast is the wood basket of the world. We produce not quite, but almost 20% of the global wood supply. And we have 50 million acres of forest plantation. But climate change is a threat. And unless we plant more trees and manage our forests better in short order, we are not going to be sustainable for the foreseeable future, and we must do something about it. And as a matter of fact, genome editing technologies provide tremendous opportunities to address in different commercial markets multiple avenues and multiple opportunities. In the timber space, think construction; in the chemical space, think biofuels; and primarily in the fiber and pulping industry, everything that's paper related and fiber related. And obviously, beyond this, of interest to our audience today is the classical canonical ag traits of resistance and resilience to pests and fungi and disease, drought, heat, cold, maybe even given the weather that we have right now ongoing, and global warming, and even viruses, as we all know, sadly. So there's a lot of opportunities to deploy those breeding technologies to breed a better forest and enhance breeding. In our case, we use CRISPR, clustered regulatory interspatial palindromic repeats, as Joerg pointed out with Jennifer's Nobel. It's a great disruptive technology. The effectors in this case are CRISPR-associated sequences, like Cas9, guided with a guide RNA that enables the very precise geolocation of an endonuclease like Cas9 that nicks DNA 3 nucleotides away from the programmable sequence. And then in our case, for trees, primarily doing knockouts, we can generate and select for NHEJ-driven repair that generate knockouts of phenotypes or genotypes of interest to generate phenotypes of interest to us. For foresters, this is important because we can reduce from 15 years to 12 months one breeding cycle, more than 10 times faster. In terms of regulation, it is safer. I hope we'll talk about the secure rule. As addressed, it is transgene free. We do so using a DNA-free RNP modality for delivery. We don't require viral or bacterial vectors. We just deliver Cas9 proteins loaded or preloaded with the guide RNA. And as mentioned with the secure rule, we do not at this point in time fall under, in the US and increasingly beyond the US, regimes that would construe those technologies as GM-based technologies, which is very important for consumer acceptance, and very important in terms of messaging to the public to embrace this technology under the guise of increasing, maintaining, and promoting biodiversity. We've been working on this company for a couple of years here in North Carolina. Again, you can see right here we have NC State red edited trees using RNPs delivered in a DNA-free modality to knock out some genes of interest that alter pathways that are physically visible here. And my colleague and co-founder Jack Wang at NC State, running the Forest Biotechnology Group, has spent years of his life improving the methods necessary to efficiently deliver those tools to stem cells of trees to get the edits that we want at a scalable pace. And as we'll hear today, it's very important not just to be able to deploy the tools, but be able, obviously, to secure the IP and have a strategic plan to have freedom to operate. But very importantly, I think oftentimes people underestimate the need to really understand the genetics and the pathways and the genomes, time transcriptomes, time environment, transproteome interactions to really inform in a multiplex manner a whole genome editing strategy to impact complex traits, multigenic complex traits that can be altered efficiently in the lead germplasm to eventually deliver tangible commercial value to impact the world. So I look forward to discussing this in the panel and in the follow-up conversation, and invite you to think a little bit more about the forest and trees moving forward with genome editing coming to a forest near you sometime soon.
Excellent. Thank you, Rodolphe. Very exciting. From 15 years to 15 months. This is quite an achievement. So coming to our next speaker to follow the C.A.T motive. So CRISPR, ARCUS, Talon. The next one would be Fayaz Khazi, and I'm very happy to introduce him. Fayaz is the CEO of Elo Life Systems. Fayaz has a Ph.D. in plant molecular genetics from Auburn University. And then he went as a postdoc to Children's Hospital of Philadelphia. And there he got really in close contact with gene editing, because he was, for the first time, he could demonstrate in vivo genome editing in mouse models using zinc fingers at that time. So it's quite some background here. Fayaz joined then Intraxon and had their multiple leadership responsibilities in the areas of human health, food, and animal therapeutics. 7 years ago, he joined Keygene as the CEO of Keygene USA and established a business in North and South America. So Keygene is a Dutch company, and he made a footprint here in the US. Obviously very successful, because Precision Biosciences then recruited Fayaz to join as president of Precision Plant Sciences. And Precision Plant Science actually was then rebranded as Elo Life Systems. And this is the company Fayaz will present to us now. So Fayaz, please.
Thank you, Joerg. Let me share the screen real quick. And let's go to full screen. All right. Thank you all. So what I want to do is maybe talk about, what Elo is in terms of a company. How do we see the space, and how are we using our ARCUS genome editing technology, which is a meganuclease homing endonuclease-based technology. So when you think of Elo, the reason why we formed this company was to be able to take both computational biology and all the things that are going on in computational biology and put those together with ARCUS genome editing technology and target both of those compelling technologies in unison towards improving the health of the people and the planet. So Precision, Elo is a local company. We are right here in Research Triangle Park. And we are a wholly owned subsidiary of Precision Biosciences. We've been doing genome editing for about 15 years now. And over the years, we partnered with multiple companies. We worked on multiple crops, and we've identified certain lack of efficiencies in when you put together genome editing, molecular biology, computational biology all together to expedite the product development cycle. We kind of figured out, okay, here's the bottleneck, here's another bottleneck. So we kind of optimized a lot of workflows, which makes us go from a whiteboarding session all the way to an edited plant in a significantly fast time frame. And all of this has been done by my team here at ELO. We are a team of 23 scientists. Right. So just to get into the size of the opportunity, several years ago, maybe 3 to 5 years ago, the industry was still wondering, whether the consumer demand for healthy food, nutritious food, natural ingredients, is that a fad, or is that going to stick around longer? And the data is here today, where this is here to stay. People are looking for better alternatives, and it's a significant market. That ridiculous number that's below there, $6 trillion, is the total addressable market. So that means it's a interesting proposition for use of technology to address these markets. So when I was at Precision Plant Sciences, we scoped out the opportunity, then we saw an interesting space. So there are the seed companies here, and then there are the specialty companies here. So then there was this space in between the seed and specialty company. And we said, this is a beautiful space for us to occupy, because we are working with the large CPG companies who have a commercial pipeline, and we can enable those pipelines. That way it is a quicker and a faster and a risk-free approach to the market for our technology and the products that are created using our technology. Right. So then we looked at, are we a genome editing only company, or are we a products company, or are we a solutions company? So we chose the latter. We chose the latter because in agriculture, as you all know, it's so complex that just one technology is not going to solve the problem. So you need to have all the components figured out. If each one of these components is available at a different location, then once again, it forms a whole different kind of a bottleneck. So we said, why don't we put all of these things together under one roof? And that's exactly what we did. We put together a data sciences team that harnesses all the DNA genomic data that's available in the plants already. Then you create the information, and that information should be converted into knowledge. And only then you can utilize the power of genome editing technology, because this is important to remember. Genome editing is less about targeting, it's more about targets. And I think Rodolphe also mentioned that on one of his slides, because that's a critical bottleneck. In plants, because there are about 350 species that are of interest, things become complicated very quickly. So then the question is, how do you bring these two together under an integrated pipeline and start making products? And at that point, another thing to remember is agriculture in general is investment heavy. Upfront investment is heavy, timelines are longer. So we said, how about we bring a commercialization partner into this mix on day one before we lift a pipette? And that's exactly what our business model is about. So our business model is flexible, partnership oriented, and investment light for the company. So there is, investment from both the partner and the company towards making a product that the partner is willing to commercialize, right? So that's the start to finish for ELO and how we work. And just to focus on the front part of it, this is the data issue. The data issue currently is, all the decisions about targets and targeting are made based on, a biased approach. Right now, it is a very manual, artisanal approach. The current data sets are sporadic, disconnected, and of relatively poor quality. And then the outputs are also very linear, very time consuming. So one of the things that we are solving for is, how do we utilize the power that's available in cloud computing, massively processing multiple data sets and generating outcomes that have a very high coefficient of predictability of that genotype to phenotype correlation. And the reason is this, right? Because precision editing or genome editing is so precise, you cannot say, here's the gene in this particular zip code. You need exactly the location where exactly it is. It needs to be find my phone kind of scenario where it says it's under the couch kind of scenario. You cannot do much with gene editing when you pretty much have, you know, a very broad, a QTL size target. So this is the pipeline that we have put in place, and so far it's doing very well. And from an operational perspective, you know, when you're looking at workflows, the workflows also need to be highly efficient. So we built all of these apps which we can just drag and drop and you know, develop or at a click of a button. Workflows that can be customized to anything that we are looking for, any question that we are asking, regardless of the genome. So these workflows are genome independent, crop independent, question specific, and you can identify or you know, have different stringencies associated with it based on the question. And then the second part also is, you know, when we are targeting one or the other gene, it's really critical to understand what the gene model looks like. So we did some innovation in this space. You know, this is the context likelihood relatedness approach that's one of the more popular ones in the academic space. And this pretty much looks like a hairball. And one of our data scientists, Angel's, worked on this from NC State. She worked on this, and she, you know, clarified the model which we typically use on a regular basis. So this has, what we've done is step by step, we made simple processes that we take for granted all the more efficient, and we are connecting each one of those you know, processes and steps in any laboratory by making it all the more efficient. So just to switch gears and get into genome editing technology, what ours is a meganuclease or a homing endonuclease based technology, right? It comes from green algae that's all around us. It's Chlamydomonas reinhardtii. And this ARCUS nuclease has been in this algae, and it has evolved with this algae for tens of thousands of years. So the very first thing is, you know, it is this technology or this nuclease has evolved in complex genomic environments, as opposed to, you know, when we look at synthetic systems or microbial systems, we believe there is a good advantage for this technology where it is more discerning, so to speak. And over the last 15 years, we've spent a significant amount of money to understand every detail of our nucleases. And one of the unique features is it's an entirely a very small protein, about 360 amino acid long, and we produce that using a in silico design and directed evolution based approach. So that means it is not driven by the target, it is driven by the protein evolution itself. So what we do is we can go through multiple iterations, and those iterations will show in the precision of binding and the precision of cutting at the desired site, and also precision, the minimum amount of or zero off targeting that we see when we use our nucleases. And here's an example that we published in Nature Biotech a couple of years ago in a non-human primate model, where, you know, with multiple iterations, off targeting pretty much was absolutely nothing in non-human primates. And since ARCUS nucleases are utilized both for human applications and slight variant protocol is utilized for plant applications, off-target analysis is a part and parcel of everything that we do. So in short, ARCUS is a nature's own genome editing system. It's one of the smallest ones that's out there among the four. It's efficient and versatile. It has very high efficacy. It gives you 3' sticky ends, and it's a product-ready technology. I can make a product today and take it to market tomorrow. And we have a significant amount of patents here for the molecule. And over the years, we have all these partnerships that we have executed upon. And one of the more recent one was a very interesting and a very nice partnership with Eli Lilly with potential to generate about $2.7 billion for the company. So we are really excited about this. And the one tidbit is, the very first genome edited crop plant was created using our homing endonucleases in collaboration with DuPont Pioneer Hybrid back in the day. So we've been doing this thing for a long time. Here are our facilities, some of our watermelon programs, tomato programs. We are in the Alexandria AgTech Center in RTP. And in terms of using the technology for products, we talked about computational platform, ARCUS nucleus platform, and our focus on multiple crops, not just one crop. We think, if you address the computational biology and the nucleases, it should not stop you from working on multiple crops at the same time. So you don't have to be an expert in corn or watermelon or banana. The technology will allow you to do it. So if that is true, what happens immediately is plant-based product becomes the common denominator, and there are all these areas for where you can truly make a difference, whether it's from a health angle, from a food angle, or from a sustainability angle. And this is our pipeline at this point. And this is a nice demonstration of our multi-crop expertise. So we have a zero melon program where we are making naturally natural products, a natural sweetener that's 0 calorie and has a significantly high level of sweetness compared to corn syrup. So this is a program that's in greenhouse trials at this point. This program to create ultra low saturated fatty acids, we started that with Cargill back in the day, and now we brought those programs back into ELO, and we're working on that. And this is the Saving the Banana, the TR4 Fusarium wilt related program is partnered with Dole. And we embarked on this program last year, and we've made a significant amount of progress. And finally, at ELO Australia, our subsidiary in Brisbane, we are working on climate smart plant-based proteins. As we see, the demand to plant-based proteins is going to increase, but right now, most of the plant-based proteins are produced in colder climates. So when you come south for the acreage, there will be a climate-related issue, and we are kind of trying to address that preemptively. And with a local North Carolina partner, Avoca Ashland Chemicals, we are working on clarisage-based production of sclereol. So where are we going with this? This is a CGIAR C.A.T created list of crops that are trending towards becoming a mainstay in the next 50 years. So as you can see, what you see is, the current staples, which had a start, maybe in the 60s and 50s, the Green Revolution crops are what we primarily eat. But these are the ones which are the opportunity crops. And being a multi-crop company, this is where we expect us to be playing in the coming years. So to summarize, there is a huge target market, versatile technology platform, and it works on multiple crops. We have a computational biology system that's tapping into everything that we learn. Nothing is going to waste. And we know that our model is infinitely scalable because we know what it takes to create a product and how many people and at what cost, right? So if you do that right, then all of these areas could be very interesting for ELO. So I'll leave you with that. And if there are any other questions, I'm available later on after 5:15 in the networking room.
Excellent.
Yep.
Thank you, Fayaz. Let's move directly on to the next one. This is Travis Frye from Calyxt. Fayaz, perfect. Here we go. And there is Travis. Excellent. So quick introduction from Travis. Travis is the Chief Technology Officer at Calyxt, and he has his PhD obtained his PhD in plant biology and biotechnology at the University of Delaware in a program together with DuPont Pioneer. So there was already a little path on the way, so to speak. He also has an MBA from University of Chicago, the Booth School of Business. And prior to joining Calyxt, Travis was worked in several large multinational agricultural and biotech companies. As there are not that many large multinational agricultural companies, you can take a guess which companies he worked for. So very much looking forward, Travis, to your presentation of Calyxt.
Thank you, Joerg. So hopefully my presentation is up and coming through. Thank you very much for having us be able to present here. I'd love to tell you the story of Calyxt. We've been in existence for about 10 years. We are a publicly traded company, and as such, I get to share this very first message from the lawyers. Just to let you know, some of the things that we'll be talking about are forward-looking, so take that as it may. But really what I want to talk today to all of you about is specifically Talens and how we use gene editing, and specifically the use of Talens in our traits that we brought to market. We're the first company to actually deliver a actual commercial product to market. And we've got another one that's getting ready to be released pretty soon, and I'll get into that by the end of the talk. The future is, as you've seen from the first two talks, are really is super exciting in terms of places that we can impact with gene editing. And I'm not going to change that story. I think we look at the world as a place with a lot of challenges ahead of us that we need to solve. And we believe that the future demands action, specifically in getting to healthy and sustainable solutions. And at Calyxt, we're delivering those plant-based innovations for a better world in a lot of these areas that I'm showing here on the slide. When you think about Talon and Talon as the system, we were very fortunate. The company was created out of the University of Minnesota, and one of the co-founders of the company, Dan Voitas, who many of you probably know, who invented Talon. He's been very involved in not only building the company, but also training the scientists that we currently have inside the walls here. So we've built a system with Talon that we're very, very versed in. We understand it very well. We understand really how to deploy it to go after the traits that we think are most important. And every day we're continuing to innovate on this technology. And so just to give a quick illustration of what this technology can do and how it works, much like the others, it's a cutting technology. And the difference with Talon over many of the others is we use these TAL effectors that are able to target anywhere in the genome. And the reason why we like Talon specifically within what we do is because we can target anywhere in the genome, including across multiple genomes. So when you start getting into more complicated cropping systems that have multiple genomes, in the case of wheat, having talents that can be specific not only to genes, but genes on specific genomes is very, very important. And as we've seen recently in Nature Communications, talents are really, really powerful when you start getting into heterochromatic regions that are traditionally a little bit more resistant to some of these technologies. So we use it primarily as a way to knock out genes. Obviously, it has other capabilities in terms of knocking in and positionally inserting genes, but we use it primarily as a knockout system. And we're working on new technologies to even modulate genes and target, almost creating, as one of my lead scientists calls it, a potentiometer of sorts, where we can kind of move beyond the on and off and more towards sort of the regulation and potentiating the way the gene is interacting within the genome. The benefits, as I was alluding to around Talon, the precision is first and foremost something we really look for. There's a lot of reasons why that precision is important. First of all, we want to hit the targets that we expect to hit. But as you bring a product to market and you bring it through the regulatory systems that exist both in the US and globally, they want to know that you're not hitting any off targets, and the only genes that you're editing are the ones that you intended to. And with this system, we have the type of precision and predictability to understand where those talons are going to interact. And if they are going to be doing any off targeting, we are able to predict that ahead of time so we can search for it and make sure it doesn't happen. Obviously, the efficiency of Talon in our hands has been really, really useful across just about any crop we've tried. We love the way that it works. We've got a lot of in-house expertise in building them and obviously deploying them. And so it's something that we then can drive ultimately the cost of the technology down because of the way we use it and the technologies and automation we've built in-house to leverage it. And then lastly, and probably one of the more important aspects of the Talon system is the fact that we have a market value case and a very clear path to commercialization of gene edited products, which hasn't always been the case for some of the gene technologies. So this has helped us get products to market quickly, which has allowed us to be the first to market with some of these technologies to get them out into the hands of our customers. We believe, and the way we run our business model is that there's a number of different areas across the industry and multiple markets that we believe gene editing can influence and impact in a positive way. And the first way we do that is working together to ideate with partners in these various areas to understand what their needs are, what they need in their new products to either be a new flavor, could potentially be something functional in nutrition, or even in the case of renewable clean energy. How do we build a product or a crop that they could use and do that from the very beginning in our ideation phase? Then we deliver those traits, or in many cases seeds, to those customers, which you'll see in a few slides in just a bit. And then they actually bring that product to market. And so we work together with these partners to go from the beginning to the end. We manage all the regulatory, if there is any, getting through the US system, and then obviously navigating any of the global that might exist. And so we've done this a number of times now, and we feel very comfortable that we can take many, if not all of these products from start to finish with the right partners. And as I alluded to earlier, we did bring the first gene edited product to market in the form of a high oleic soybean oil. This was a really exciting product that's on the market. It's doing really, really well. We've made some announcements recently of larger ADM actually taking on a lot of our grain this coming year to produce this oil. So it's really exciting. And the idea was we were able to bring something that was a more sustainable high oleic version of oil that was something that it was a cleaner taste. It worked functionally better in the systems it was being used in the frying systems because it provided extra frying life, which allowed less waste to be accumulated. So from a sustainability point of view, that was a really great benefit. And then just the nutritional benefits that came along with having a high oleic soybean oil in the market. And then lastly, one of the things that was really exciting was the fact that we could locally grow and process a product that restaurants love to put on their table and cook their food in and deliver that to their customers. So it was a great message to show a locally grown and processed oil that was on the market. And then the latest project that we're really excited about is a collaboration that we did with S&W Seeds, and specifically in alfalfa. Really what we were trying to do here is help them deliver a higher digestible performing product for livestock feed. And what we were able to do is take their industry leading high yielding varieties of alfalfa and then actually edit and improve the digestibility of that. And so now we have a really high yielding, high digestible product that they're able to sell on the market, which helps to reduce the amount of alfalfa needed to produce the same amount of milk or protein. It obviously reduces the amount of waste that's coming out of that cow because they're able to produce more while eating less. And then obviously transportation costs and water use across the whole system is improved. So we're seeing a really nice sustainability story here with a product that ultimately was something that with one partner had all the pieces they needed from a yield and a performance agronomic perspective, but needed to just improve a small component around the digestibility, which was really hard to do through traditional methods. And so this is where gene editing was really powerful. And with that, I'll point anybody else to some of the other products we've got on our pipeline. If you want to go to our website, you can check us out. And I also will be around after this session to host a small Q&A at the end. So I'll turn it back over to you, Joerg.
Excellent. Thank you very much, Travis. And with that, I think we're ready to go into the panel questions. I've seen we have already a number of questions piled up from the audience. That's great. Most of these questions are actually going into the regulatory consumer territory. Of course, I have prepared many questions on the technology and business, so we will probably cut that part a bit shorter and directly start then earlier with the regulatory consumer questions, because I think this is what most people are interested and like to hear. But nevertheless, I think this is such an exciting technology and also the businesses we heard now. So let me start off with a couple of questions in that area before we go then to regulatory. So Rodolphe, maybe starting with you. You founded a number of different companies, many of them not in the agricultural area. So what you see is the specific technology need for agriculture with the gene editing tools you are using, and maybe you have some ideas where this might develop in the future.
Yeah, I'm going to echo a comment I made earlier and a comment that Fayaz agreed with. It's really informing the targets, not the targeting anymore, for your genome editing strategy, right? Up until recently, up until the last few years ago, targeting was hard. It was difficult. It was a bottleneck. At the advent and the advances in genome editing technologies, or the ARCUS, TALENS, ZFNs of CRISPR, have kind of transcended that challenge. So that's great. So now the bottleneck is really, if you want to multiplex that and do it a bunch of times, if you're efficient enough and precise enough, assuming that we are, then what does your list of targets look like? And to answer your question, Joerg, compared to what we know in human genetics, our knowledge of plant genetics is only partial, right? Let alone tree genetics, as I mentioned for me. So we know between 100 times less or 1,000 times less about plants and let alone tree genomes than we do about human genetics. And we still know how hard it is to cure genetic disease in humans. So on top of that, the conditions are very multiple and variable, right, in plants across the globe. The genotypes are more diverse, arguably in many ways, than human genotypes are. So the genetic complexity is not really accounted for and is somewhat of a barrier to go past your top 5 or your top 10 candidates. And this is where really, as Fayaz and Travis mentioned to some extent, complementing genome editing tools with good genomics and big data and good understanding and machine learning based predictive models that enable us to not have to do it all but pick the winners ahead of time is going to be really a game changer. We're not there yet. Some companies are, some companies are working on it. I mean, I know we are big time. But I think this is really where, when you compare to therapeutics and we compare our genetic knowledge overall, having solved, quote unquote, the cleavage targeting part, really informing a sustainable pipeline of gene targets for a multiplex editing strategy is not quite where it needs to be for most in the industry.
Excellent. Fayaz, what's your take on what would be your next technology wish in that field or need actually?
Yeah, and I fully agree with everything Rodolphe said. The only other thing that I would like to also add is in agriculture, partners, people, investors become impatient very quickly. They want to see results much faster, and tissue culture is another major bottleneck because the current mindset in tissue culture is from the past, because there was a reason why tissue culture is practiced in a certain manner. But now that you can create all these variations so quickly, there is very little patience to wait for 9 months to see a tiny plant show up phenotype, right? So I think that's where, and I don't think it's going to be a huge issue. It just needs to be a concerted effort and a matrix-based testing, and we will get there. There are quite a few advances being made, both in our company and elsewhere in protoplast biology, protoplast transformation, regeneration. It needs to happen more and more.
Travis, anything to add from your end?
No, I would say I echo both Rodolphe's and Fayaz's comments. I mean, targeting is a lot easier now with the technologies. I still think at the end of the day, it's what targets you go after. And we're still very early on in terms of understanding, we're still in that on or off. I mentioned earlier, one of my lead scientists really wanting that potentiometer. That also is being able to, rather than just turn on and off things, but it's being able to understand the smaller gene effects that interact and regulate. And so you can get into more complex types of traits, which are really where a lot of our partners are wanting us to go. So that's something that we're building into our systems to be able to do that. So that's kind of the next big wish list, is how you get to that finer resolution of understanding of the targets.
Excellent. So maybe staying with you, Travis, a quick fun question. We had the cat in the egg, Dr. Seuss. What's your favorite Dr. Seuss character?
Oh, wow. I would, there's so many of them, but I mean, outside of I would have to say it's, outside of the cat, I'd have to say it's the cat, right? I mean, he's the best one of all.
Okay, the cat learned a lesson. That's the message. Excellent. So I'll get another question for you on gene editing. Travis, is kind of, you mentioned it with the alpha in your presentation, gene editing as a technology also to tackle some of the major challenges. Here in this case, reducing methane production, CO2, greenhouse gases. So what other opportunities do you see to tackle these major challenges with this technology? We talked before about finding targets. It's difficult, but maybe you have some ideas. And I will also ask, of course, then Fayaz and Rodolphe.
What really excites me is the fact that we have an opportunity to really leverage the full power of what plants have to offer. I think all of us, at least on the call here, got into this business because we love plants and love plant science. And, plants have been giving us life for a very long time in terms of the way they support our ecosystems and they support us with food. And, everything's driven from that plant. And so I think when you look across the industry, there's a pile of industries that aren't using that potential and aren't tapping into it, don't even know that the potential exists yet. Areas, in renewable plastics, packaging, even some of the advanced materials sectors out there. There's a lot of potential for different plant materials to be used in different ways than they currently are. And because it's so much a part of who we are as an ecosystem. And so we believe at Calyxt, allowing plants to see their full potential is something we want to enable. And obviously Talon is one of those tools we're using and refining to help us do that.
Yes.
Could you repeat the question? Just
yeah.
The last
just,
part of it.
I mean, you mentioned also in your presentation the idea of tackling the global questions. You know, your topic with the zero melon was more in terms of food health, but what other topics do you see at the horizon for tackling these really big challenges? Yeah.
Absolutely. Thank you for that. I was chatting with Rodolphe and talking about his favorite Dr. Seuss character, and that's when I missed your question. My apologies. I think, you know, the mission of the company is, you know, we identify this, you know, what is that one space that currently is an opportunistic area for us where technology can make a difference, right? So what we are trying to do is do more in terms of solving the lifestyle diseases, so to speak, right? So look at lifestyle diseases and how can everyday diet matter? And so why aren't there, you know, solutions for lifestyle diseases? Because it's a cumulative effect. So how do you keep certain things that we are genetically predisposed to at bay, even for 10 years? It would make a lifetime of a difference, right? So that's number one. The second one is, again, there are crops that people know how to grow. There are crops that are economically, you know, make a lot of sense to produce. So then the question becomes, and it's an interesting possibility, is what more can the crops do? Is watermelon only a red sweet fruit, or can it do more? Because, you know, it doesn't take a whole lot to grow a watermelon. So we want to take genome editing beyond the knockout kind of solutions. And I think we talked about that a little bit. So there are these nuanced approaches where you can take a commodity crop and elevate that to a premium crop, exactly like what Calyx has done with the soybean with the high oleic trait, right? So that's how we look at it. But the bigger problem that we are trying to solve is, you know, connect the nutrition part to human health. And number two, look at, you know, assisted migration of crops in response to climate change. So what can we do to preempt what's coming our way, right? So those are the major areas that we are focused on.
Rodolphe.
Yeah, interesting question. So I'll stick to the trees just for the sake of the argument here. So, you know, we all hear about, you know, planting 1 billion trees, planting 1 trillion trees, and nobody's saying which kind of trees are where, right? So it's like people saying to feed the world, we're just going to plant more corn, and we're going to plant 1 billion acres of corn, and we're going to solve the problem. And it's just so naive to me as a scientist and as a business guy that you really have to have a plan, right? Which environments and what species and what biodiversities and how you're going to manage the forest. And so if we really want to combat, you know, global warming and be more sustainable, how to manage, you know, non-arable land, and how to pick, how we can make the best use of, you know, water and resources and soil and biogeography and biodiversity to really strategically plant forests where they need to be and where they can impact the environment and where they can impact us. And then I would love to get to a point, as just discussed by Fayaz, of, you know, can trees be more than just carbon capture? Right? Can trees be more than, you know, capturing CO2 and you know, slowing down the pace of climate change? Because there's a lot of trees that feed us, right? There's a lot of trees that have a lot of attributes for chemicals and biochemicals and nutritionals, you know, and more sustainable biomaterial, as Travis said, right? Think of like turning cellulose matter into bottles to replace plastic. I mean, there's a lot of possibilities. And the challenge is people are not, because they're impatient. Again, I agree with Fayaz, and the loan investors looking at timelines of trees. If you think they're impatient with crops, you can imagine how impatient they would be with 15 plus years of cropping cycle for trees. So I think really having a long-term perspective on big problems like this is important, because it does take time. It is difficult. It does require, you know, tissue culture and big data and on and on and on. And you just have to commit to doing this. And as long as we're short-term minded, short-term sighted, it's going to be really hard to address those grand challenges. And I think we have to be strategic enough, committed enough, wise enough, invoke the wisdom of the forest to really commit to having a long-term impact.
Very good. We talked already about business targets, and I'll start with Fayaz now. And before we go, you also get a fun question. So we know Rodolphe's number plate, you know, from the movie, or some people saw it on the campus at NC State. So it's CRISPR. So Fayaz, what do you have? ELO 1 or? You have something fancy? If not, you don't have to say, but I'm just wondering.
No, mine is something HAL, HAL, maybe based on the movie Shallow Hal. I don't know.
Okay. Very good. No, excellent. So the question I have for you, and this goes around what we also discussed before, is and we heard it several times, is how do we pick the targets? You had this nice comparison saying, look, I mean, if I want to find my phone, it doesn't help me to tell me what zip code it is in there. So if it's under the couch, I need to pinpoint it. So with that regard, we talked a lot about the power of the technology, different tools there. But how do you see the interplay between technology push and really business pull, where you say, look, what are the targets? Are we really looking for real business cases, or are we trying to, you know, use the technology to direct it some of these topics?
Yeah, no, that's a great question and an important one for us, because our entire thesis around ELO is founded upon that question. And it is, you know, we don't want, you know, we are scientists at heart, but the consumer is not a scientist. And the age of technology push, we have seen how long it took to, you know, control the damage. So at this point, the way we centered the business model as the core of our company. So that means we are addressing only those things where there is and some sort of a benefit to the consumer immediately, and it's obvious, and it's not to a niche consumer, but to the bigger mass consumer. So one way for us to do that is, you know, my team of 23, no matter how hard we work, we won't be able to get the right kind of consumer information and market data. So we said, let's open up the business model where we recruit the companies on day one and say, hey, first question, should we work together? If the answer to that is yes, half the times the answer is no, we don't care. But when the answer is yes, we say, what is your number one on your wish list? Because once you know what that number one product concept is on your wish list, then as scientists and a technology-based company, everything is easy, right? To them, to those companies, the hard part is the science. So we say, we don't talk about the science, we say, this is what's going to happen, right? So for us, every program that you see has been a technology pull. The most you know, popular example is TR4, you know, when you see the headlines, banana is extinct, right? So it's a dramatic headline, but it is a technology pull because livelihoods, economies, industries are at stake, right? So whether we will be phenomenally successful or fail miserably, that's to be seen, but we are working very hard to be successful, right? So that's just one example. The Zero Melon was also a technology pull. It came from one of the largest beverage companies, and they said, we wished we had something that was not super expensive to produce, and it was produced in the mainland in the US, and we didn't have to export something, you know, like monk fruit sweetener import monk fruit sweetener from elsewhere, right? So these kind of things, I think, matter quite a bit, and I think Alan has a series of comments here, Alan Wenck. I'm looking at it, and I think that's one of the key parts. Technology pull versus technology push is going to determine the future of genome editing companies and genome editing products, at least in the first phase. You train the consumers, you train the regulatory agencies, and then you take it to the next level. At least that's how we are looking at it.
Rodolphe?
Yeah, I would echo again what Fayaz just said, and some of the questions that have been in the Q&A, notably Alan's. I think we have to learn from previous shortcomings, right? And PR in the 80s and 90s was terrible. And today we live in a context where scientific skepticism, alternative facts, anti-science bias are real. So we have to be even more cautious and mindful of developing narratives that are compelling, not just to the scientific community, not just to the investors, not just to our strategic partners, especially when they're a good fit, but really engaging with regulators and politicians and the public. Because the technology can be the best technology in the world, but if there's no excitement and acceptance, we're not going to be able to unleash it and deploy it and implement it. And whether it's, again, ARCUS or CRISPR or TALENS or CFNs or any gene editing tech. And in ag, it's even more complicated compared to therapeutics, because saving lives is an easy pitch, right? You take a sick patient and you make them, you save them or extend their life, or you solve their problem. In the case of doing that into plants, crops, even livestock, somebody asked about livestock online, or even trees, it's a lot harder to get the narrative from the recipient because they don't talk, right? You can relate to them, you can put some good music in the background, you can have the nice visuals and everything, but it's not like a patient telling you they're gonna die if you don't treat them and they don't take your product. So altogether, when you look at all those challenges, really being more strategic and wise and more proactive on the PR side is extremely important. And that's something where ag has great stories to tell, but we don't have a lot of good narrators and necessarily narratives that are polished and ready to go. And I think storytelling to relate to a diverse audience, again, to a point that was made, because now we're in a global world more than ever. There's all kinds of opinions, all kinds of concepts, all kinds of biases and preferences and inequitable we need to account for. And really having a polished PR piece, a team of 23 people, it's hard enough to do the science, forget about the rest. So, you really need to work as a team. I think collectively we need to work as an ag community to advance that, commit to this. And this is where universities like NC State University, can really, as part of their mission, make themselves available to allow the ag industry to convey message of responsible research and innovation and transparent science management for the betterment of humankind and the planet. And I think there's a lot of work to be done there. Again, we have, it's on our radar for a lot of people, but we have so many business problems to solve in the short term that oftentimes committing to PR can be a hard to justify item, but extremely important for eventual success.
Excellent. Perfect. We have reached a Q&A time frame. I think we can try to connect to some of the questions. We already started with the PR topic. I want to start with Travis on this topic of, and this was coming up a couple of times in the questionnaire. It's kind of like, it looks like that the US regulatory system is favorable for at least single mutation edits, which are not considered as GMO. But globally, this is a little bit a different picture. So how do you see, you're working with soybean, this is a global traded crop. How do you see that globally, and what are topics like traceability and these factors?
Sure, it's a great question, and you're totally correct. I mean, in the U.S., the regulatory framework is pretty well understood and spelled out, and we were very specific in the way we managed the crop as it was going through, and actually after it went through the regulatory sign-off, because it's not obviously an approval process. It's sort of an acknowledgement that yes, it is a non-GM. It doesn't have any foreign DNA. And it was naturally derived, genes, and the genome was where it was supposed to be. Once you show all that, then it's a matter of managing that in the US. Obviously, depending on where you want to take the product or who may take that product further, then yeah, the regulatory agencies in other countries, other locales, some have a little bit more advanced. I think most recently we've seen Japan just approved GABA tomato. There's a few other countries that are similar minded, I think, or I should say similar process to what the US is. And then there's other countries that are a little earlier in their process development, right? And we're working with them because we've got some of our products going through those approvals, or at least we don't necessarily call them approvals, but through the process so they can understand. And to your point, a lot of it's education. So, just defining what it is. There is no foreign DNA. There's no new, DNA being put in. There's no new proteins being introduced. So, yeah, it's partly education and just allowing the agencies to figure out what they want to do and giving them the information they want to make the right decisions. So.
Very good. Going back to Rodolphe. Rodolphe came off cheap so far. He didn't get a fun question. So Rodolphe, you get also one fun question, even if it's not in the Q&A. If gene editing would be a food or a seasoning, what would it be for you?
Oh, it had to be French wine because it's going to get better as it ages. And everybody needs some to be healthier. No brainer. Have 1 cup every day. You'll be better off for the rest of your life. So
excellent.
I like the trick question for sure, but that was an easy answer, I guess. Yeah.
Very good. I'll come back to what you said before about stories, and we had that also in the question. Is kind of like, how do we tell the story to the consumer and do not get into the same trap we have gotten with previous technologies in that area, where there was a very strong reluctance then to take that up. And so for consumers, it's not easy to understand. Travis said, yeah, there is no foreign DNA in there. Sometimes maybe people don't even understand what DNA is. So what is your story you would tell, and how would you advise to move in this topic and being proactive with communication and education?
So great question. And again, touching on a lot of the things that we went through, and I'm going to go back to being facetious like Joerg here. And the answer was in the slide you presented with the Human Nature movie poster. So I think my answer would be, trying to tell people what we're doing or how we're doing it is irrelevant, right? Unless they want to, get a CRISPR class at NC State University, or go to a seminar, or get a slide deck about genome editing and DNA sequences and the like. Rather than the what and the how, it's more the who is doing it and what motivates them. And that's why stories like Jennifer's story, again in the movie, or Walter Isaacson's book on Jennifer Doudna's life is coming out next month. So like the who is doing it and the why they're doing it. The who makes it relatable, because we're people, we're consumers, right? Like I eat, I hope I'm going to get maybe not a bottle, but a glass of French wine tonight. And we eat the food that we make all day, every day, right? So why do we spend our lives and our careers and work hard to solve those problems, right? Because we have the same issues and concerns that all consumers have, because we're consumers too. What motivates us, what drives us, and what makes us relatable to consumers? The movie does that really, really well. So I like that in that sense for sure. And then the bigger question is the why. We're not creating problems, we're providing solutions. So we're not there to play God and have superiority complexes, academics and whatnot. We're trying to feed the world, have more sustainable agriculture, and solve climate change. And unless, I think this is what I said in my opening to some extent, grand challenges to really be solved require us to deploy grand solutions and techniques. If it was easy and all it took is plant a few more crops here and plant a few trees there and drink a glass of wine, we wouldn't have the problems that we have. So we really need to harness the most powerful technologies that we have. And again, people understand that in therapeutics, unless we have diagnostics and a vaccine, we're not going to solve our current pandemic problem. But guess what? It's the same when crops get viruses. It's the same when trees disappear because of infectious disease. We need to use the same best-in-class technologies and solutions to enable their own genomes to fend off infectious disease and to become more efficient and sustainable. And breeding and evolution is nothing new here. We've been farming for millennia. Just unbeknownst to us, we didn't know at the time how to genotype the crops that made it. And so I think that focusing on the whom and the why more than the what and the how is really how we're going to solve that narrative problem.
Excellent. And just to put a scientific number behind the recommendation from Rodolphe on the Human Nature movie, it has a 97% on Rotten Tomatoes, so it's definitely worth to look at it. Looking at the questions, the high rank question was on what regulatory frameworks were used to evaluate Talon for off-target implications? Travis, maybe you can have a shot at that.
Sure. I mean, I think I'm not sure frameworks is really if that's really what, I'm not sure how to define that, but what we look at when we target a trait itself is we look at what the Talon is actually going to land on within the genome. And then because the size of Talon is large enough that it's very precise, right? It tends not to jump around. If it does, we can predict where it will go. And so in most of those cases, we design the Talons to be very specific so they don't jump around. There are cases when we run the risk that it could be a Talon that might land somewhere else, but we know where it's going to land. So then we look for that in the back end to make sure that doesn't happen. Part of the process of going through the regulatory hurdles is to show you don't have any off targets. The genome is clean and free of any foreign DNA and all the things you need to prove that you're a non-regulated article under the current regulations. And so that's what we go through for each and every product. And in the US, that's still, it's voluntary, but we do it for all of our products, just as a matter of fact, so.
Excellent. Another high rank question, and this goes to Rodolphe for TreeCo. The obvious targets are enhanced biomass or speed to maturity in order to address sustainability. Are these the obvious targets?
So I would argue that there may be some of the targets, but I don't think they're the best ones. As a matter of fact, the chemical composition of wood is very important for efficient processing. So by changing lignin content, lignin composition, SG ratio, lignin percentage, and things like this, you can really breed wood to be more amenable to processing, to be more efficient. And the wood processing industry, as a matter of fact, is not the cleanest industry whatsoever in the world. So you can really have an impact on energy costs for processing of wood. Imagine a mill that does 1 million metric tons of wood processing per year. Black molasses and energy constraints and inefficiencies in enzymatic pathways and heat control. There's a lot of things we could do genetically that's relatively straightforward that can enable us to make wood more efficient in terms of processing, and that they can debottleneck capacity and increase that. If we think about sustainability, I think water usage and disease resistance is very important, right? If you're going to plant 1 billion trees, you're going to plant 1 trillion trees, picking the right traits in the right environments that are hypervariable and changing and dynamic and susceptible to disease, whether it's fungal or viral or bacterial or any combinations thereof. There's a lot of pests out there that are canonical traits in the ag world that we need to address that provide sustainability for longevity in terms of the lifespan of the forest. So there's a lot of targets that we could look at. And then after that, it's industry specific as well. So if you use wood for furniture, the sandiness, the texture, the fiber, even the color can be edited at one of those things on my slide. So so you can have pigment-free colored wood. Imagine not having to paint the wood and toxic paints and chemicals. So there's a lot of things we could think of on the spot here that all contribute individually, in toto, altogether, to a much more sustainable set out there. And there's, again, and don't forget, most people don't know, I didn't go through that, but the average tree genome is about 10 times the size of the average human genome. And so there's a lot more targets to pick from that we need to work on.
Yeah, just following up on that question, I think the next highest one rated is also very interesting in terms of our regulatory discussion. It goes again to you, Rodolphe, because I think everybody's wondering what would happen if we take that wood and export it to Europe, to maybe Germany, who is not so keen on anything genome editing. Maybe France would be even better nowadays, but let's take Germany. How do you see that? Because this is very interesting. Nobody will eat wood, not that I know. So it's kind of like comparable to maybe cotton, but you also know that cotton has also some history. So maybe you have an opinion.
I mean, absolutely. So I think the consumption part of the consumer is important. So if you have, genome edited wood, that's furniture, right? That's not alive. That's not gonna, pass on to the next generation of tables. I mean, this is like, a wooden table, there's another wooden table. So I think consumers understanding what they do with the product can make them more comfortable. You don't have to eat the wood. Most people should not eat the wood, right? But by definition. And I don't know how secure or insecure, even in Germany, people will feel about the furniture that they're using or the cardboard boxes that their Amazon Deutschland packages get delivered through. So I think people don't realize some of those products that are derived from woods. There's a lot of paper, the paper you print on, and if it's more sustainable, then why not? So I think educating the benefit from a consumer standpoint, where you don't eat it, you consume it, but you don't consume it orally or intravenously or others. I think there's a lot of opportunities to have a conversation there to make people more comfortable with the acceptance of something that they have a mental blockage about because they don't understand it, they don't trust it, they don't rationalize it. But when it's tangible and something they can touch and put in their hands without having to consume it, I think we have opportunities to increase their tolerance and rethink what they think about this. And to your point about how quickly the world is changing, I mean, I still can't believe 3 weeks ago the French Minister of Ag stated in public setting in the news, quote unquote, NBTs are not GMOs, right? Novel biotechnologies encompassing CRISPR are, in his opinion, not GMOs. And I think the world is about to change. Because if we live in a world, in a panel, we say, maybe even the French, despite their wine, may be open to rethinking how they perceive genome editing and reassess what's going to happen next with the European Court of Justice efforts underway and some of the great narratives we're going to get from the great German ag companies that have a lot at stake and great narratives to tell. I would forecast boldly to some extent that it's a question of when rather than if mindsets will change. And within the when part rather than if, I would even ponder how soon versus how late. Again, I'm an optimist. We'll see what the Lorax or Dr. Sue is having in the cards for us moving forward. But I'm cautiously optimistic about mindsets changing, even in Europe, even in Germany, dare I say.
Excellent. The breakout rooms are coming up, but I want to direct one question to Fayaz, because I think we have here one which is also fairly high rated and very specific. So let's address that, and then let's see if we get remaining questions in the breakout rooms. So this is about the TR4 banana project. How will you breed for sustainable resistance? Are you targeting single genes or multi-gene approach? are you accounting for differences in fusarium genotypes? So very specific question, Fayaz.
Yep, and because it's so specific and it's a partnered project, I really cannot answer that without getting into trouble. But I can address that, in generalities. Any project, whether TR4 or any other disease resistance, right? Initially, what you would want to do is look at the regulatory opportunity, right? So you look at the regulatory guidelines and then look at whether or not there is an editing strategy that fits in SDN1 or SDN2 or SDN3. So based on that, as any program would go, you would have plan A, plan B, and plan C. So we would approach the point is to save the banana plants from the fungus that is going to kill and devastate nations, right? So that when you think about that, everything else becomes secondary. We can figure that out. That's how it's been looked at. But I truly cannot go into our strategy specific to TR4.
That's fine, and
Yeah.
I think Jean will understand that, and she can always contact you, and
Yep.
maybe there is also some knowledge at NC State for further going. So with that, I'd like to thank you very much. You three were fantastic panel here and going through my fun questions as well. And I'll give it back to Michelle.
Well, Joerg, you haven't answered. Who's your favorite Dr. Seuss character?
The Grinch.
You didn't think you were going to get off that easy, did you?
I'm the German. For me, the Grinch is the favorite.
Okay, we could all go with that. Well, I want to reiterate what Joerg just said and really thank all of you for an amazing panel and presentation. We've been getting nothing but compliments so far. And I know there's a lot of questions that maybe we didn't get to, but I still want to invite everybody to join us in the networking rooms. I've put the link in the chat. So it will take you to a different website or to a different call. So hold on a second, we're almost done. And I also want to thank our partners at Ag Start and Ag Launch and remind everybody on March 17th, we will be having our next National AgTech Forum hosted by Ag Launch, where we'll be talking about soil health. So again, thank you, everybody. And we will have a recording of this event today up in the next couple of days online. So thank you all, and we will see you soon. Take care, everyone.